research, and we're all eagerly waiting to hear them. But before that, we are privileged to have Dr. Axel Brockman with us. Axel is an eminent researcher on bee biology, honeybee biology, so to say, and it's been working in NCBS in Bangalore for the past quite a few years. So Axel will, of course, have the inaugural lecture. May I have Smita Krishnan on behalf of the, to introduce Dr. Brockman on behalf of the Indian Pollinator Initiative, please. Over to you, Smita.
Hello, everybody. It's a great pleasure for me to introduce our speaker, Axel Brockman, who would be giving the inaugural talk about studies on the behavior and biology of Asian honeybees. He has been working on honeybees for over 2 decades, starting with his PhD, which he completed in 1999 on neuroanatomy of the olfactory system in the European honeybee, Apis mellifera, following which he worked as a postdoctoral researcher in Germany on behavioral aspects of the European honeybee. He also worked with Professor Gene Robinson at the University of Illinois on molecular mechanisms of honeybee foraging behavior. Although he's been a faculty at National Center for Biological Sciences, Bangalore since 2012, he has been associated with Professor Raghavendra Garadkar's lab since 2003. So it's been a long association with India. Currently, he is an associate professor at NCBS and has continued his work on molecular mechanisms of honeybee foraging and dance behavior and biology of social bees, but of course of Asian. He would be presenting his research on drone olfactory system and sex pheromone communication, behavioral maturation in Apis cerana, Apis florea, and annual colony. I now invite Professor Axel Brockman. Studies on the behavior and biology of Asian honeybees. Axel, please.
Okay, thank you.
Parthiv, I think.
Microphones. Everyone except the speaker must mute your microphone, number 1. And number 2, should you have a question, I'm sure you will have a lot of questions, write them down on the in the chat box. And like Dr. Axel Brockman will also have, he has his presentation in 3 different segments, specific segments. At the end of each segment, should you have any specific question which you must ask at that point, kindly put that down in the chat box, and I'm going to pick that up and ask. Is there a way of muting, kind of universally muting? Just count for that option. Yeah. Please mute. Please mute. Mute all. Yeah, got it. And Axel, now please unmute yourself and speak.
Okay, yeah.
Thank you. Yeah, that's all.
Thank you, Putative, and thank you, Smita, for the nice introduction. And I have decided to give you 3 examples of the research on Asian honeybees that we did in my lab, or that I even did before I came to India, to demonstrate you what kind of questions I'm interested in and what kind of techniques I'm using. In the short time, kind of like, how to say, I have to skim over the different topics and cannot kind of delve into deep detail. So, and I just give you the highlights, and I hope that kind of like you still kind of like understand the project and the question. So, studying behavior and biology of Asian honeybees. So, let me introduce you that the latest phylogeny of
recording in progress.
Latest phylogeny on the honeybees published by Deborah Smith last year kind of suggests that there are 15 species. And without going into detail of all the 15 species, kind of like we see a major separation between open nesting honeybee species that belong to giant honeybees and the dwarf honeybees that build one comb, and then the swarm and of the colony kind of aggregates around the comb, builds a curtain to kind of like protect the brood for predators and environment brain. And then we have a large group of the cavity nesting honeybee species, which actually kind of nest in a cavity like a tree hole. And in addition to living in the cavity, the nest is constructed of many multiple combs. All of the honeybee species, except one, live in tropical Asia, tropical Southeast Asia, including India. The only exception is Apis mellifera, which is distributed from Europe to Africa. In India, okay, wait a minute, sorry. In India, there are about 7 species living in India. And let me just introduce you to them. Among the dwarf honeybee species, we have Apis florea, and Apis florea is kind of distributed all over India. And we have the sister species, Apis underuniformis, which is mainly kind of occurs in the northeast region. And has a kind of in the northeast region. Then we have the giant honeybees, and everyone knows Apis dorsata. Apis dorsata is distributed all over the Indian continent, whereas the sister species Apis laboriosa is only occurring in the Himalaya. The molecular data kind of published by Deborah Smith kind of indicate that Apis dorsata occurring on the Indian continent might kind of be composed of populations of two species. She suggests that there is a South Indian Apis dorsata population of species. And then we have the cavity nesting Apis. The new phylogeny suggests that there might be 2 species, kind of like Apis cerana and the the yellow Indian Apis cerana. So everyone knows that Apis cerana is kind of coming in the black strain and then the yellow strain. Now, Deborah Smith suggests that these might also be 2 species. Independent of whether these are species or populations, with 7 honeybee species, India is one of the countries with the highest diversity of honeybee species. And because of that, actually, it is a country kind of like well suited for comparative studies of honeybee biology. What you have to, so what we know about honeybee biology is mainly based on the study on Apis mellifera. If you kind of look at the publications, about 95% of all the scientific publications are on Apis mellifera. Whereas kind of like we have only little kind of like knowledge about the biology and the behavior of the Asian honeybee species. Because the honeybees are important pollinators in Asia and tropical Asia, I think, in my opinion, it's important to study the biology and behavior to conserve these pollinators. So there's, how to say, so we need, so we have to increase our studies. Unfortunately, kind of like in most agricultural universities, people are studying Apis cerana, or they are studying even Apis mellifera. And that is because you can do beekeeping with them. But so it's only, we have only one species or two species with which we do beekeeping. All the others are wild. And so we have to actually take care of them and study them in the wild. As mentioned by Smita already, kind of like I want to introduce you the way I do the research. And you have to understand that I started as a neuroanatomist. I was interested in the brain. And from that, I went to behavior. And then in the US, with the sequencing of the genome, I started working on molecular mechanisms underlying behavior. How we do that, I will demonstrate now in these three topics. So I will talk about the drone olfactory system and sex pheromone communication, behavioral maturation, and the latest is about the annual life cycle of Apis dorsata colonies. To understand kind of like what I'm talking about now, it is that a small introduction. So if you compare the head of the drone and the worker, you see that the drone has enlarged eyes and actually also has enlarged antennae. The function of the drone is to mate with the queen. And so the drone enlarged its visual system as well as its olfactory system to kind of like detect and kind of find the queen. And for the olfactory detection, kind of queens and drones use sex pheromone. The queen is sending out a sex pheromone and the drone is highly sensitive to the sex pheromone. And you can see that they have an enlarged antennae, and then they have, compared to the worker, specifically increased the number of a specific sensillum that is composed of a sensory neuron sensitive to the pheromone. And if you see the difference, it's about kind of like 9 times more pore plates than workers. Now, in relation to this morphological dimorphism, if you look at now at the olfactory receptor genes that are expressed on the antennae, then what you see is that kind of like there are specific olfactory receptor genes that are more expressed in the male antennae. And there are also kind of some olfactory receptor genes that are higher expressed in the female. Now, regarding the male biased kind of olfactory receptor genes, these are probably kind of candidates to perceive the sex pheromone. And in a study in the US that I was involved, we could identify that the highest expressed olfactory receptor gene is actually detecting 9-ODA, which is a major sex pheromone component. Now, during my PhD, I actually got already involved in studying Asian honeybees. And the initiation to study Asian honeybees goes back to when I read a paper on the phylogeny of the genus Apis. And in the phylogeny by Alexander, one trait that differentiated Apis florea and the dwarf honeybees from all the other honeybee species is that they had a short male antenna compared to long male antennae in all the other species. For a person that is involved kind of like in the study of olfactory communication and perception, that kind of like, that difference kind of like suggested to me that there must be a difference in the olfactory system. Here see you the scheme of an Apis florea drone. And here you see two pictures that kind of like the same size on Apis florea drone and an Apis mellifera drone. And here you can even kind of like see that kind of like Apis mellifera has hugely enlarged antennae compared to Apis florea. And kind of the enlargement is kind of like over proportional to the increase in the head size. So Apis florea has kind of large eyes, but has kind of like smaller antennae. And if you kind of look kind of at the antennae and kind of count the pore plates, then we have the 18,000 pore plates on the drone antennae of Apis mellifera, but only 1,400 pore plates in Apis florea. That's a tenfold different, and this tenfold different means kind of like a strong decrease in the sensitivity of these antennae. What I have to say, what you cannot see here, is that although the Apis florea drones have only 1,400 pore plates, there's still a sexual dimorphism between workers and drones. The workers have only 600 pore plates. So, so there's still kind of like a sex pheromone olfactory system. When I came to India, to NCBS, that was around 2011, the genome of Apis florea was sequenced. And a colleague at NCBS, Professor Saudamini, and me, we decided to annotate the olfactory receptor genes and study kind of the differences in the expression of drone and worker antennae. So as I told you before, kind of like in 3 slides before, this figure here shows you the differential expression of olfactory receptor genes on the drone and the worker antennae. To the left, you see kind of like the male biased or drone biased expression, and on the left, on the right, you see the female or worker biased expression. If we look at the drone biased expression, we see here in blue, these are olfactory receptors that are already kind of also higher expressed in Apis mellifera drone antennae compared to worker antennae. And among those is the olfactory receptor number 11, which is the one that detects 9-ODA. However, in addition, what we see is that we find a high expression of an olfactory receptor that is called OR155. So, florea drone antennae and mellifera drone antennae kind of like differ in the number or in which olfactory receptor genes are higher expressed. And here to the right, you see a comparison of the major olfactory receptors higher expressed in the drone antennae. And the figure shows you the fold change difference between drone and workers. So how to say, so you see here that, so here this OR11, that is this 9-ODA receptor, which is hugely increased in Apis mellifera drones compared to the workers. It's not that much increased in Apis florea. And different to, as said, different to Apis mellifera, we see in higher expression of this olfactory receptor gene 155 that does not show a male bias expression in Apis mellifera. The interesting thing is that the olfactory receptor gene 155 belongs to a family that is supposed to be detecting floral components. In parallel to our molecular studies on the drone olfactory system, a colleague in France, Bastien and kind of Jean-Christophe Sandoz, worked on the differences in the brain. The first area in the brain that is receiving the olfactory receptor, the axons of the olfactory sensory neurons is the antennal lobe. And later a student of Joby will talk about the antennal lobe in Ibis dorsata, I think. So, but so this is a scheme of an antennal lobe of a drone, an Apis mellifera, and a scheme of the drone antennal lobe in the drone of Apis florea. What you at the moment need to know is that the antennal lobe is kind of composed of ball-like structure. The number of the ball-like structures correlates to the number of olfactory receptor genes. So, and we know that this green one is actually detecting, and in the male system, what we find is that there are a few of these glomeruli that are hugely enlarged. And these are kind of important for the perception of sex pheromones. And we know kind of like, for example, that this green kind of macroglomerulus is detecting 9-ODA. And kind of Bastien et al., they could show that that there is a homologue macroglomerula smaller in Apis florea. So Apis florea is detecting and using a 9-ODA as a sex pheromone. But then we see kind of like different other macroglomeruli in Apis mellifera compared to Apis florea. Based on these anatomical differences, we would suggest that Apis mellifera uses more components for sex pheromone communication than Apis florea. The interesting thing here is that there's evidence from morphology and from molecular biology that the sex pheromone communication system between Apis florea and Apis mellifera are different. Based on the molecular studies, Bastien came up with an evolution of the sex of the dawn antennal lobe system. I don't go into detail, but how to say, so what we now know is that there's a high degree of variation in the components, probably drones used to find the queens. And that there are changes in the components that they are using, and there's an increase in the number of sex pheromone components that they are used for communication. And then, kind of like as shown in Apis mellifera and Apis florea, there's probably a high difference in the sensitivity toward these components. Traditionally, sex pheromone communication and sex pheromone system have been studied in moths. And in moths, you don't see this variation within a genus. In moths, kind of like the sex pheromone communication is very similar. Kind of the variation, the probable possible variation in the sex pheromone communication in honeybees is huge compared to other groups. And I think it's an interesting system to study why that there are so many differences. And then you have to realize that this kind of phylogeny doesn't even include Apis antenniformis, a sister species to Apis florea, Apis laboriosa, a sister species to Apis dorsata, and kind of like yellow cerana. So that was the first part. Is there any question regarding that part?
No, there isn't.
Okay, good. Then I'm continuing. So behavioral maturation in Apis florea and Apis cerana. Behavioral maturation in Apis mellifera, or kind of like division of labor in honeybees, is based on behavioral maturation. And this has been studied in very great detail in Apis mellifera. And kind of like how to say, so if the worker is young, it does cleaning the cells. If it's getting older, then it's starting to feed the larvae. Then it's starting to feeding nest mates. And in later age and mid-age, it's kind of like involved in packing pollen and also kind of in comb building. And at the age of 20 or 25, the worker starts foraging. And then it still lives 14 days. So division of labor is based on these, what we call behavioral maturation. Behavioral maturation means that the animal shows different behavior at different ages. This is kind of like a general phenomenon. And we also see some kind of like behavioral maturation. Of course, we see also behavioral maturation, In humans, kind of like from 5 to 20 years, I mean, we have puberty, so we don't have sexual behavior in the young children, what we call children, and kind of sexual behavior kind of like develops over time. So behavior maturation and the brain processes underlying behavior maturation are kind of widespread in the animal kingdom. However, one of the best studied kind of like examples of behavior maturation or behavior ontogeny is kind of like behavior maturation with honeybees. And Gene Robinson, kind of where I was a postdoc, his lab was kind of one of the major labs studying behavior maturation in honeybees. The idea is that this process of behavior maturation is regulated by hormones, and these hormones kind of induce kind of changes in the brains that are based on changes in gene expression in the neurons, and that these changes kind of have something to do with the change in the behavior. And in honeybees, kind of like the major hormone that is kind of inducing this behavior maturation or regulating this behavior maturation is juvenile hormone. And what you see here is that around the age of 10 days, juvenile hormone kind of strongly increases in the honeybee workers. And it's kind of, and we think it's a pacemaker of the behavioral maturation to become a forager. Now with the genome sequencing, kind of Gene Robinson's lab looked into the brain and looked kind of like whether there are changes in the gene expression. And what they could demonstrate is that there indeed kind of changes in gene expression. Here it kind of like shows you a PCA of different groups. So the A is basically a worker that has just emerged from the cell. B is kind of like composed of workers that are about 4 days old. C are kind of like a gene expression pattern of workers that are kind of 8 to 20 days old. And D is kind of like the gene expression in forager brains. So it's kind of with the increase of the juvenile hormone, kind of we see a change in the brain expression. And then we see also correlating with that, we see a change in the behavior. Now, my postdoc, Shruti Krishnan, she looked at the behavioral maturation of Apis florea and Apis cerana, of an open nesting species that kind of like has only one comb and where many kind of like workers sit in the curtain, compared to a cavity nesting species that doesn't have a curtain but combs. So first, she looked kind of like, I'm going a different way. So we're starting with the behavior. And so I have to, I'm sorry, I have to explain. In Apis florea, in Apis cerana, we can observe the behavioral maturation as we can do it in Apis mellifera, because we can use an observation hive and we can look at kind of like the bees, what they are doing. In Apis florea, we cannot do that because kind of like the comb is hidden behind the curtain and you cannot kind of open the curtain. So the predictor of behavioral maturation here is the onset of foraging. That is what we can see. We can mark bees, introduce bees into the colony at day 1 and then see when they become foraging. And what you can see here is that, and I'm starting with Apis cerana. So Apis cerana, kind of most of the workers become at age of day 20 foragers. 60% is because you are losing kind of introduced workers during the experiment. But this becoming forager at an age of 20 is very similar to Apis mellifera. However, if you look at Apis florea, they have a totally delayed behavior maturation onset of foraging compared to Apis cerana. And it's kind of like, if you look at 50% of the introduced workers becoming foragers, there is about, Apis, in Apis cerana, it's about 20 days, and kind of like in Apis florea, it's about 70 days, almost kind of like 4 times longer. So, and that is a huge difference. If we look into from the phylogeny, and kind of like Apis florea is at kind of like it's a more ancestral species, then one has to say that Apis cerana workers show an accelerated behavioral maturation. Now, then, Shruti, kind of as I told you, this behavioral maturation is based on kind of like juvenile hormone titers, and kind of like, so she measured kind of juvenile hormone titers using mass spectrometry in bees of different ages and kind of bees of different tasks. And here I show you just a figure about the difference of JH between workers in yellow and foragers in green. And what you see is that juvenile hormone in both species, in the open nesting as well as in the cavity nesting Apis cerana, are higher in the foragers compared to the workers. Then we look at the genes. And so these genes, kind of, I don't want to go into detail, but these genes that are here kind of shown are transcription factors. So in transcription factors are actually initiating the expression of other genes. So the idea is that the hormone of the juvenile hormone kind of like is bound by a receptor of the brain neurons. And then there is a second messenger cascade that initiates the gene expression. And kind of like the ones that are expressed at first are transcription factors. And what you can see here is that kind of basically that in most cases, the green, the foraging related transcription factors are also higher expressed in the foragers compared to the nurses. What does that mean? So we think that, and this is only kind of a few data of the data set, is that actually the major hormonal regulation as well as the gene regulation regulating behavioral maturation is the same in Apis florea and Apis cerana, and probably Apis mellifera. So the regulatory toolkit or mechanism or skeleton stays the same. But then we still have to explain why is the onset of foraging kind of accelerated in Apis cerana and Apis mellifera compared to Apis florea. And our hypothesis at the moment is that either kind of like in Apis cerana, kind of like we see an accelerated increase in JH compared to Apis florea, or we see kind of like a lower variability in the response thresholds in Apis florea. Something I haven't talked about and what appears to be getting interesting is the function of vitellogenin. Vitellogenin, it also works as a hormone and it regulates JH. Actually, in the nurse state, it inhibits JH, and kind of like and when JH increases with age, JH kind of stops VG expression. And what I haven't told you is that we have some evidence that vitellogenin expression in Apis florea is extremely high. So it might be that actually the regulation of the pace of behavior maturation is regulated by changes in the vitellogenin control on JH. Okay, so that was about the hormonal regulation of behavior maturation. Is there kind of
no more question?
Okay.
There's no question.
How much
I have a couple of them, but I can ask them.
Good. How much time do I have still?
You have another 10 minutes.
Okay, thank you. Now I'm talking about the annual life cycle of Apis dorsata colonies in Bangalore. But to understand kind of like the annual life cycle of Apis dorsata, I first introduce you to the annual life cycle of Apis mellifera, carnica, that means the European honeybee. And here you see a scheme of the annual life cycle. And how to say, as I mentioned, kind of like as kind of 95% of the literature on the European honeybee, people think this is the annual life cycle of most of these honeybee species. So how to say, because you have the winter and snow, during the winter, Apis mellifera kind of like builds a winter cluster and does not fly out. And then kind of before spring, kind of like the queen starts to laying eggs, and then the colony kind of increase to grow. And in kind of the temperate zone, kind of like the major flowering is in spring. So and how to say, and these colonies kind of like strongly increase. And here's the size, and they need to increase the size, producing workers to be able to swarm and do reproduction. And kind of like at late spring, early summer, kind of like we see kind of like the reproduction and swarming. And the idea in Apis mellifera is that the old queen stays, the old queen leaves the colony and the new virgin queen stays. So swarming kind of like leads you to two colonies. And then, so by summer you have two colonies, and then these two colonies have to kind of like to do two things. They have to grow in size, and they have to collect food to kind of survive the winter. And this is here kind of food collection for the winter. And then kind of like before kind of like an end of fall, kind of like the queen stops laying eggs. And the winter is the highest selection pressure that these colonies appear. If you don't have the size to kind of build a winter cluster, sufficient big winter cluster, and if you don't have collected sufficient food, you will die during the winter months. Now we started studying annual life cycle in Apis dorsata. That is a study of my student Rajat S. And we started this by observing kind of like the presence of colonies in a kind of like a residential compound called Bhatia city in the north of Bangalore, kind of like close to NCBS. And kind of like this the chair contains 10 towers that have up to 26 or 30 floors. And this map kind of shows you where we found bee colonies over the years. And the interesting one is the yellow ones, which shows kind of nesting sites at that area that were kind of like occupied by different colonies over the observation time. So you still see that there's preferred nesting site, and then kind of colonies, other colonies kind of go to other sites. The numbers indicate the floor. And okay, so and here you see kind of like different figures about his observations. So he started in 2018 until 2020, and then came the pandemic. And this figure shows you the absolute number of colonies present in that compound. And what you see is that, let's start here. Of course, during the monsoon, the number of colonies is low. And then after the monsoon, it increases, kind of like in November, December, and January. Kind of in November, December is the first flowering season. It kind of like goes down here in this one, but I'll say this might be especially here. And we see kind of like a little kind of increase in April and May, and then it goes down again. during the monsoon. So this is kind of like known, and there have been other studies in Bangalore that kind of like Apis dorsata colonies are kind of leaving during the monsoon area, and we think that they do a migration to some other destination. However, it's an interesting result is if you look at the arriving and departing colonies per month, you see actually that, and here it's kind of arriving is in blue and departing is in orange. You see even in phases, kind of like in the non-monsoon phases, actually kind of like a lot of colonies are departing. And here you see in April kind of like an increase. This is probably not arriving. That might have something to do with swarming. And that the swarms are kind of staying at that nest site for some time. And then kind of like you see that, okay, there's increase in departing bees during the monsoon, but still during the monsoon, kind of bee colonies are arriving at that place. So underneath kind of the cycle of total, kind of underneath the cycle that we see that the total number is decreasing in monsoon, we see kind of like at a frequent arriving and departing. And it's clear here if you look at the duration of the residency. So, and here what you see is that actually most of the colonies only stay for 1, 2, or 3 months. And that means kind of like, how to say, that is even smaller than the good flowering season. So the consequence is that we think that so there are 2 kinds of migration. One is kind of like the monsoon migration, and then kind of like in a shorter time period, the colonies are relocating them. And so the dorsata, so the dorsata colonies are highly migratory. Sorry, that is. Okay, let me kind of cannot move. Let me see. I'm sorry about this. Oh no.
Maybe you go out of
no, that's a problem.
Full screen mode and then
Kind of yeah, let me kind of sorry about that.
That's all right.
The next slide is a little bit heavy, so let's see that it works now. Okay, good. Sorry about that. So now, kind of like how to say, based on our observation, we come up with a first model of the annual colony life cycle of Apis dorsata. So let's start. Where do I start? I start kind of like at the end of the monsoon. So at the end of the monsoon, colonies are coming back, and that is kind of like October, December. And if the colonies are coming back, the first thing that they have to do is build the comb. And then they are kind of starting, and after they've built the comb, they are starting to produce brood. And then it appears that kind of how to say, what they can do is they have started this process, but for whatever reason, kind of like the nesting sites might be not as good as before, and they decide to relocate their nest, and they're migrating somewhere else. We don't know for how far they are migrating, but in Apis florea, there has been demonstrated such a behavior also of nest relocation. And they think it's more like in the range of a few kilometers. At the new nesting sites, they kind of like, again, need to build the comb and then start producing brood. and kind of like to get in kind of like the phase of swarming, the colony has to kind of like that, you know, from Apis mellifera, has to have a specific size. And then kind of like, if it has a specific size, then the colony starts kind of producing sexuals. And that is kind of like the beginning of the swarming season. And then kind of like, if you look at Apis dorsata, kind of hard to say, and Apis florea, so it appears that kind of like swarms are leaving that colony, but different to Apis mellifera, actually kind of like they leave the whole nest. So there is no kind of part of the colony that stays with the old nest. And then kind of like you have these new colonies that is kind of like in April, May. And then kind of like, how to say, the colonies have to, they might also still increase, but then before the monsoon, they again need to stop laying eggs and prepare for the migration. And then we see this migration. And kind of at the moment, my pet hypothesis is that the seasonal migration is actually kind of very similar to the winter class in Apis mellifera. And there might be then also reproductive diatoms. But so what we observed last year was actually that not all, and you saw it also in the figures, it is that not all of the colonies are leaving Bangalore. Some kind of like even stay during the monsoon. And for one colony, we observed that that colony that stayed during the monsoon in Bangalore, that they showed actually a swarming season in October and December. So different to Apis mellifera, it appears that Apis dorsata, for example, doesn't only have one swarming and mating season, but two, can have two. So this is what I wanted to show you. And sorry, oops, the last slide. No, wait a minute. Is that now these studies of observing colonies in Bangalore kind of lead to a project that is in collaboration with Smita and with a Japanese colleague and funded by Google AI for social good. And it's called Exploring and Managing Human-Bee Conflict in Asian Cities using AI. The goals of that project is kind of like what we want to kind of have is try to get is an estimate of the population size of Apis dorsata colonies in Bangalore, and then have an estimation of how many of these colonies are killed. So the other way around, kind of like the project started by the common procedure of humans in Bangalore that they ask people to remove and kill the colonies that are kind of nesting on balconies. And the question is whether kind of like, how to say, this kill, what is the extent of this killing of Apis dorsata? And does this killing of Apis dorsata actually have an effect on kind of like the pollination service of these colonies? To estimate what is the percentage of the colonies killed, we need an estimate of the population size in Bangalore. So, and this is based on long-term monitoring colony abundances. And for that, we want to develop a citizen science project, I've watched, so that kind of people can kind of observe bee colonies for us. And we kind of like, for the killing of colonies, we want to kind of perform an online survey with the apartment complexes. There are about 800 apartment complexes, to kind of study how much of bee colonies are there and how much they are killed. And kind of like how to say, we want to interact with the public by having a web page in where we kind of like show our results. And finally, because we want to understand the pollination service of these dorsata colonies that are living in Bangalore, for kind of like possible agriculture, we want to kind of like study, I mean, we want to find out what is the destination area where these colonies that are kind of nesting in Bangalore during the monsoon. And that is kind of like a first part of kind of studies, kind of like in the conservation of Apis dorsata colonies. So that was what I wanted to show you. And I thank you for your attention.
Thank you very much, Axel. It's now open for questions. Quick couple of questions if you have one ready.
So,
no, directly, I mean, I guess one needs to write the question in the chat box. If you can quickly write them in the chat box, and then that can be picked up for, yeah.
So these are not just the questions, but that's just a bit of the supplementation.
Supplementation, I'm sorry, it can come up at the end of the presentations.
Okay. It was nice listening to Dr. Brockman.
Yeah. We can definitely enrich ourselves with your supplementation at the end of the session, because there are 5 students we have to hear them. So if there is no question, is there a question? Is there a question that can go directly into, yes. So Sachin, where do the Apis dorsata go during monsoon migration? This is one question.
Yeah, I think we don't know. Maybe there are people that have some opinion about it, but I think it's actually not known. And yeah, so we have to study. I cannot.
Okay. Anyone else? I have one I was writing, but then yeah, a quick one. This difference in the olfactory receptor in between florea and cerana, which you show, does it really impact the population size? Because that's a very crucial thing. A drone's ability to find the queen, you know, will that impact the overall population size of the of these 2 species?
Yeah. So the question is, so how to say showing that there are differences, and one can ask, as you asked, why is that, right? And the only answer at the moment that I have is that, how to say, we are starting always with Apis mellifera, and then we think that Apis mellifera is kind of like what we find for Apis mellifera, what's for the others. And actually, Apis mellifera is the outlier. It has the largest antennae. So all the other Asian honeybee species have also, they have the large type, but they have kind of smaller antennae than Apis mellifera. And my idea is that, how to say, you increase sensitivity to the female if the density of your mates is low. So it means, kind of, how to say, that probably the density of colonies, kind of like in temperate zone, because of, also maybe because of nesting sites, kind of low abundance of nesting sites, is lower. So they have to fly and they have to find a queen.
Larger distance.
Area, right?
So it's kind of a selection pressure on, working because of the habitat sort of harshness or whatever.
Yeah. So in the other way around, kind of like how to say, if you have a, so we don't know what this sensitivity means, but on the other hand, you can also say that, for example, what we see in Apis dorsata congregation, and there have been reports that also the distribution of Apis florea colony is patchy. So how to say, so kind of like a stay kind of like closer by, and then you also get it regulated.
Right. Okay. Thank you very much. There is one quick question from Dr. Bhatt, who says, do we call it, why you call it a diapause? If you want to take that, Axel.
Okay, good. It's kind of like how to say, I'm not a good entomologist. So reproductive diapause, what I mean is that the queen stops laying eggs. Yeah.
I think we are good with that. I thank you very much, Axel. And yeah, we need to move on now.
Okay.
And we have our first student speaker here with us, who's okay. Sandhya is from University of Hyderabad, and she's again going to talk about the Apis dorsata olfactory behavior. And may I have Sinu to kindly introduce the speaker, please. Dr. Sinu from Kerala Central University.
Thank you.
Kindly, introduce.
Yeah, thank you, Dr. Putative. So we are moving on to the student seminars. We have 5 speakers. The first speaker is Sandhya Mogli. She completed her MA in English and was teaching in engineering colleges before joining Jogi's lab in University of Hyderabad to study the oscillations in olfactory discrimination in bees. She has taken up his dorsata as a system. Over to you, Sandhya. Thank you.
Yeah. Sandhya, you can now start to share your screen, please. Yeah.
Good afternoon, everyone. I thank the organizers of Pollinator initiative for giving me this opportunity to present my work. It's titled An Investigation into the Olfactory Discrimination of Role of Oscillations in Olfactory Discrimination using Apis as a model system. As we know, honeybees are quintessential pollinators. They're also very useful as model behavior repertoire. Can you hear me?
Yeah, it's little patchy, but yeah.
The connectivity is poor.
Yeah, please go
on. The honeybees are amenable to a variety of assays, among which the proboscis extension response, where a hungry bee extends its proboscis when presented with the sucrose reward, is widely used. This proboscis extension response, or PER, can be conditioned similar to the conditioning of the Pavlovian dog. When a hungry bee is presented with an order stimulus, forward paired with the sucrose reward, after a few learning trials, the bee extends its proboscis to the mere application of order, having associated the order with food presentation. So, Brickman et al. Have developed a training protocol where a bee is presented in 3-4 seconds of order, overlapping with 3 seconds of sucrose presentation. This is a single learning trial, and there will be a few learning trials based on our requirement. And between each trial, there is a time interval, which is called the inter-trial interval, or the ITI. In every trial, if the bee extends its proboscis before the presentation of sucrose, the bee is considered to have acquisition. And after the training trials with a memory duration, the bee is tested by presenting the order alone. If the bee extends the proboscis for the order alone, the bee is considered to have memory for the learning. If the bee does not extend the order to an untrained order, the bee is said to have discrimination. So we use this paradigm to study olfaction, the role of oscillation in olfaction. If you look at the olfactory system of insects, antennae of the insects contain hair-like structures called sensillae. These sensillae house the ORNs, or the olfactory receptor neurons. The ORNs have olfactory receptors to which the odorant molecules bind. This chemical signal is transduced into electrical signal, and they are transmitted to the primary olfactory center, or the antennal lobe. In the antennal lobe, the ORNs synapse with the local neurons, or the LNs, and the projection neurons are the PNs. The local neurons are restricted to the antennal lobe, whereas the projection neurons innervate the higher olfactory structures, the mushroom body and the lateral horn. In the mushroom body, the PNs synapse with the Kenyan cells, or the KCs. So if you look at the connectivity between the ORNs, LNs and PNs, the ORNs have excitatory effect on the LNs and the PNs, and the peons are inhibited by the LN activity with the GABAergic action. So when a repeated stimulus is presented, the peons are excited with the ORN input and inhibited with the LN input. This repeated excitatory and inhibitory input causes synchronous activity in the peon, which results in the synchronous activity received by the KCs. So the local field potential, which is a measure of summation of population activity or the synaptic input received by the KCs, exhibits periodic oscillations. It was shown in locusts that with repeated order presentation, close to 30 Hertz frequency oscillations are present. These oscillations are also reported in moths, honeybees, and other insects. These oscillations are abolished by the application of phytotoxin, which is the antagonist for GABA receptor. But the exact role of oscillations in olfaction is not yet known, and the investigations are on. In a landmark study, it was shown that if bees are treated with pictotoxin, it impaired their fine order discrimination. In this experiment, they trained 2 sets of bees. One set of bees, the brains were perfused with saline. In another set of bees, the brains were perfused with pictotoxin. Both the bees were trained in the similar conditions, and both conditions, the bees showed good acquisition. But when they tested for memory, the bees trained with saline, treated with saline, could discriminate the fine similar order from the trained order. But the bees treated with pictotoxin could not discriminate the similar order from the trained order, but they did not have a problem discriminating a totally different order. From this, it was concluded that abolishing oscillations impaired fine order discrimination. These experiments were done using Apis mellifera. Though they are very good with behavioral experiments, there are few bottlenecks when it comes to electrophysiological manipulations. So we try to check if
your voice is breaking.
Apis mellifera, Apis dorsata, the Indian bee, could provide manipulations.